topo plot can display as well the time series, keeping the spatial location, and as well as source estimation directly on the anatomy. The workflow usually for when you have data to process with Brainstorm is to have the source data will be the anatomy and the sensors, and they go through the registration process inside of Brainstorm, and from there also we have the recordings, and using the recordings and the co-registration we can compute the source estimation, and from there we can continue with the analysis. That will be as simple as averages. We can do group analysis, time frequency, connectivity, and others. So what I'm going to show now is how it is done on a single subject. The idea is to three main blocks: that it's importing all the raw data, preprocessing, and then to analysis of the data. Well, here in the database, we'll have these buttons that they represent that we want to see the data, the anatomy view. So we have all the anatomies files per subject. So this is just a single anatomy for a subject that can have different modalities. Then we have recording that will be different sessions for the same subject. That will be the second view. And the way data is organized in Brainstorm is we have first a protocol. And inside our protocol, then we have a subject and a condition. All the data that is saved in the database is saved in the hard disk drive in the same structure. So it's possible to find it very easy, the equivalent between each other. With the original files, they are not copied directly to Brainstorm, but they are read as in the fly, depending as they are needed. This allows to not have superload, to not copy data when needed. We can optimize as well the reading functions instead of having to load all the data on memory. So the basic registration of Brainstorm is done through the fiducial markers. That those are marked in the anatomy, and as well those are obtained from the functional recordings, EEG and MEG. At the moment they were where the sensors are, the location of the sensors, and these can be done either by in this case of EEG with a standard position of the electrodes, or it can be used with devices like the Polhimus driver in order to digitize all the locations for sensors and coils. Some of the basic quality control of the signals will be to perform the power spectrum to see if a channel is completely off, of the chart for it, and check as well the interference of power line. Bandpass filtering as well. We can inspect manually all the recordings to find electrodes or segments that are bad, that they need to be rejected. So they will be ignored when we are doing averaging. Brainstorm has several features in order to detect automatically blinks or to detect automatically heartbeats, and this can be used later for the artifact rejection removal. Well, regarding just a bit note of artifacts, they can be well defined, like heartbeats or blinks, or they can be other artifacts that are not as reproducible. In the first case is the one that they can be modeled, and we can use it to be removed from the signals. In the second category, usually a way to deal with them is just to mark those segments as artifactual and reject those. For electrodes for artifacts that are stereotypical, what we can do is to detect them, then to use different methods like PCA to find their special components, and to remove those components and reconstruct our data without the influence of those artifacts. This is just one example of the. We have a cardiac artifact that is shown in the MEG signals. Then when we detect them, and we align them, we can see that this is the influence that of the ECG signal on MRI of MEG. Then we just cancel that component. We come back, and then we have our MEG signals without the influence of the heartbeat. Same thing can be done with links. And we have different methods for the composition that implemented in Brainstorm that are like popular in the literature of EEG as well as the MEG. There are a few features in Brainstorm that allow the detection of the automatic detection of artifacts. I'm going a bit quick over this slide so we can jump to the hands-on. This is the specific case of the electoral devices. Sometimes we have these squid jumps that are automatically detected. We have these jumps in the baseline of the signal. It's automatically detected and removed. About the different types of experiments, there's only two examples that we can analyze: resting state or event-based. That is the data we're gonna analyze today later. Again, and about this is on regarding triggers, how triggers can be read. They can be read directly from the data. In the case there is a channel that is digitized specifically for the trigger. Otherwise, they can be read from digital channels that were recorded either with the presentation of the stimuli or were recorded with digital inputs in the EEG or MEG device. Or sometimes what we do is we have as well the analog signal, and we can detect the trigger as well there. And Brainstorm provides all these tools to detect triggers in different modalities. Once in the case of event-based experiments, where we have read all the triggers, what we can start to do is we can cut our signals in those specific segments, and like with sometime before the event and sometime after, and these are the segments that are called epochs in Brainstorm. And through epochs, we can as well sort them. If we have epochs where there are overlapping segments, or will be ignored in the further processing. And Brainstorm allows us well to explore the data in different ways. We can explore all the sensors, the same trial, as well. We can explore all the trials for the same sensor in different modalities. It will be time frequency or time series. Regarding the optimal length of an epoch, that will depend absolutely on the design of the experiment. So what we want to do is to have as much data as possible without having to do the experiment too long for the participant. Other processing that is done during the important epochs are to remove the DC offset, as well to the trend and this resample. Once we have obtained all our epochs, probably the first analysis we want to perform is to average them to see the ERP or the event-related potential or the event-related field in the case of MEG. For averaging data, it's for EEG usually it's a common practice to average data from different in different runs if they belong to the same electrode, but for MEG is not the case because the head may change the position inside the MEG. So one of the techniques that is used is to use the MEG signal to estimate sources and to average the source space. And this is come back to what was Richard presenting this morning, where we have the sources over in the brain that are happening, and we have our forward model, in this case G, and that gives origin to the sensor, to the signal that we're measuring in the sensors. But as we don't have access to the source space, what we want to do is to solve this inverse problem, to be able to infer what was the source activity that gave origin to the activity recorded on the skull. This is again just regarding the inverse model. Yeah, a few slides that were not expected. And as well, one of the analyses that usually we have 15,000 sources per in the surface. We were analyzing the time series of those, of each of the vertex may not make sense. So in Brainstorm as well, we can use cortical parcellations that are well defined, that can be imported as well from BrainSuite, and to analyze our data really in average or aggregated per regions of interest, or that they're called scouts in Brainstorm. For the time frequency analysis as well, we can perform it in the sensor space as well in the source space. And the methods that are implemented in Brainstorm is the Morley wavelets, the Hilbert transform, and the short time Fourier transform as well. And with this, there are like methods as well to analyze PAC inside of Brainstorm. Connectivity metrics. Again, those are available in different spaces. They can be sensor space. They can be like a scout space. They can be like even different modalities. Like for example, we may want to compute coherence between EMG signal and EEG. In the case that we have in the medial nerve stimulation that we're going to do, the data we're going to analyze this afternoon. And this is just a bit of regarding
one just to show another one. So most of the analysis that I want to show this is for single subject analysis. And mostly today we will do single subject analysis. We will not do group analysis, but we want to show so that in Brainstorm we can perform group analysis where we can have multiple subjects, and then we can do some statistics at group analysis, get some significant results rather than single subject.
Mm-hmm. Yep. The idea is to do the analysis as we saw up to now for every single subjects, and then if we're analyzing in the source space, it would be to project them to a template, and then to analyze all the projections.
If that's some extra slides that
the extra slides
or show you.
Well, this is regarding about the Brainstorm code. Is possible to develop your own Brainstorm code, and to add there are several examples that we have. The code can be added into Brainstorm as plugins, or we offer an API well documented how to read data, how to do the different operations, how to call other processes that are already present in Brainstorm. There are a few examples of contributions to the code, and everything is on GitHub. Up to this year, we have more than 40,000 users registered in the Brainstorm forum. And we have more than 30 hours of self-training program with tutorials. We have in the website as well. We have some videos on YouTube for different workshops. There's the Brainstorm forum, which is quite active. So a lot of users posting their requests and posting their questions about how they are analyzing how they're analyzing the recordings. And this is a short list of many people who collaborate in Brainstorm. This, if you want to take the time.
Yeah, just one more slide. So this is just an advertising for another workshop that we are going to run next April. So it will be in Park City in UT, but it will be dedicated for AI and epilepsy and neurological disorders. So just another workshop that will be more specified. So for the session today, it will be just half a day. So it will be just general introduction to Brainstorm and hands-on, and show you how you can use it for one participants. We will go through all the steps that Raimondo showed. Yeah.
Mm-hmm. Good question. Yeah, while you're covering that, is it possible to show us the command line?
Yes. Yeah, I think we have the program. I think
yeah, we have it, but it's closer to the end when we are talking about the scripting.
Okay.
Okay.
Is it while you do something on the GUI, is it possible to go to background MATLAB and see what is the translation of what you just did, or equivalent to what EEG EEG Lab?
Yeah, there's not a feature exactly as in EEG Lab, the history. It's not available in Brainstorm. However, what we do is that when the users start to prepare their pipeline, there is a button that says export. So what it does, it creates a script that does exactly that. However, every single click is not being recorded, but that's part of the future development that we're working on. Yeah.
Yeah, this for today. We are here, so we'll go through the program. In the page of the program, we have mostly 4 sessions. So first one, the introduction that Raimondo did. So there is some extra slides. So they should be hidden, but they were here. I want to use them to show you some other feature during the presentation. But there is mostly 5 sessions. So loading the anatomy. So we show you how to load anatomy and how to co-register the subject and the channel and the channel information. And then we do some processing. So second step here. And then we do some analysis at the sensor level. And then we do the forward and inverse model as a monitoring feature. And then we do some analysis at the source level as well. We will show you some feature of the camera for a single subject. And at the end of the session, we will show how to do scripting. So mostly all the tutorial today, we will show you how we can generate the script and then we can run it for one subject.
Also, people giving the workshop is Tac Farinas, myself,
Uji is here.
Chinmay, Jason, that is not in the slide.
Yeah, Jason. And we have Richard as well. And I think John also is online.
I don't know how to use this software.
Don't show that.
Sorry. It's great though.
And regardless, and I believe this is the last slide we have. This said is about what data we're analyzing. Is median nerve stimulation in the right arm. Therefore, what we are expecting for now is to have activity in the left motor cortex, the left sensory motor cortex. The experiment has around 330 repetitions, only for one subject. All the MRI has been already pre-processed with CAT12, so we won't spend time. It's possible to call, for example, CAT12 for even free software directly from Brainstorm, but that will take a lot of time. So in this case, the data that was distributed for the workshop is already pre-processed. And as well, we select this specific data set because it's we have simultaneous recording between MEG and EEG. For MEG, we have 160 cardiometers, and for EEG, we have 41 electrodes. So the idea now is that because they are simultaneous, and we can create we can analyze the event-related potential, the event-related fields at the same time, see that their topographies, and we will be able to reconstruct the sources from both modalities and make a direct comparison. So yeah, that's a very interesting data set. And And as well, you can find the information in the brainstorm page. If you go to courses, you will find the LA workshop with the information. I'm pretty sure everyone has already
Okay, I
seen
can
this
do it from
page.
the- just when you go to the main page.
Yeah.
When you go to Brainstorm, so
Brainstorm.
there is the first page, new course, cutting EEG garden. Or
Oh,
can you
wow.
go, yeah, maybe,
Yes.
yeah,
Yeah,
you can
this
go
is
to
easier.
program here. And then you will see what we are going to do this afternoon. And also, there is all the step. Scroll down the menu, yeah, so this is all the program that we will go through step by step. But importantly, there is this PDF at the end. Yeah, detailed step by step.
This is the
Yeah.
walkthrough PDF that we have some physical
Yeah, we
copies.
printed some physical, but it's better to have it in computer.
So yeah, well, this PDF that will be showing the steps we are, so in case you have question, you can refer to it, if not to us. So we can have a nice space for the workshop.
So basically for each step, Raimundo or I will show you how to do it on the screen. And then we'll give you some time.
Yeah.
About the time that is displayed on the right side, so you can reproduce it on your laptop. And then we are working around all of us. So if you have questions, you can just ask, and then we can show you how to do it in your computer. So the main objective today that you will be able to reproduce everything in your own laptop.
All right, so it's does anyone is having problem with their installation to have the workshop data?
Other question, anyone is ready to start? Okay.
I have one question. You talk about the polymus using that for registration.
Yes,
A lot of
yes.
times with EEG, we don't have that information. How do you co-register without having some point?
So yeah, so for the data that we have here with the
Yes.
the
I
polymus
understand.
is already co-registered within the channel file. So when you import the data, so we will see the polymus points that we are required.
Yeah, but what I'm saying is if I have data where I don't have that information, how do
So
we then?
we have already brainstormed some caps, like standard caps for, because they are, hopefully you will be using the 1020 system.
Right.
That's how they are.
Okay, so you just have to have the file that corresponds to the cap. that
Uh-huh.
you're using. Okay.
Yes.
If you have that coordinate data in EEG lab, again, sorry, I'm just trying to translate.
Yeah.
Can be exported into here.
Important.
Yeah, exported from
from
EEG lab
EEG
into, yeah,
Yeah.
into,
Yeah, it's possible that to obtain the positions of the electrodes.
okay.
Yeah, like for the, as well, I didn't mention that in the slides, but also Brainstorm supports bits. So it's possible to read data directly from that it's in bits format. And as you mentioned, the EEG lab, I'm pretty sure it can export in bits format for in the dot electrodes TSV files.
Yeah, okay. Yeah,
And
yeah.
that one is possible to read in Brainstorm. All right.
So if there is no question for the technical side, I think Raimundo can start for the first step, and then we can start. No question.
So at this point, you should have 3 folders in your desktop. One that is called the workshop LA. Black that will have the files that were downloaded, the dataset. Well, there is a Brainstorm DB that should be empty. And the Brainstorm 3 folder that has the Brainstorm applications. So there you will need us to run that. You open MATLAB. And you just run the Brainstorm. You just run Brainstorm. I will show you the GUI of Brainstorm. If you can scale it, like for example, there's a lot of lag here. Then file and edit preferences, and here you can change the interface scaling. Is it a good size for everyone? This on my screen, or should I do it bigger?
You increase it. Increase it.
Bigger would be nice.
How about now?
Is it fine for everyone?
Yes.
Oh, right, so. Doc, you want to share some of
Uh,
the documents?
so we didn't print for all, but if someone needs the PDF, the paper formats of the PDF, we can.
Thank you.
Anyone else need it? Okay, we might need it. Yeah. What? We did it. It's fine.
Right, the first thing we're gonna do is to create our protocol here for people. Probably is empty in your side. In my case, I already did the workshop, so we will go to file, new protocol. And then we just put like workshop. AAA. There are two questions that are made here, two decisions regarding the default anatomy. In our case, we have the anatomy of our subject, so we say no, use the individual anatomy. And similar thing for the channel. We know the location of the channels for this specific subject, so as well we click in no. Those are as well the default options. And we click on create. This will give us a protocol that is empty. What we want to do now is that we have different views. As I mentioned in the presentation, we have the anatomy view, where we are going to see all the anatomy data from the protocol. We have the functional. Yeah, so what we're going to do now is to create a subject. So we do right click on the protocol, we go to new subject, subject one, and again, even appears the proper options that we're going to use the individual anatomy. So we just click on save. We'll create a new subject. And now for the subject, we want to import first of all the anatomy. So we go right click on the subject and we click here in anatomy folder. Please do not click the auto because we want to do it. We want to select some parameters by ourselves now. So for the anatomy folder, and we select the anatomy folder in the workshop data set. It's important that we need to select the file type is cat12, because this data was pre-processed with cat12. And we click on open. We're going to use 15,000 vertices for our cortex surface, and it will start to load the MRI as well to load all there are some anatomical atlases. But so yeah, we need to define our fiducials. Here we have the label for the nasal.
That's a question.
Yes.
Are you expecting everybody to be following along with what you're doing?
Yes.
So,
All right, so yeah,
perhaps we should get people up to that point.
All right, so let me go back to
So if anybody need help, if they can raise their hands, there's people here that will help.
So here we're on field the creation of the subject, so we haven't done anything yet. The protocol and the
That's the.
subject will do a right click, the subject, import anatomy folder. Is the first one, do not use the one that is auto, I think that's why your SPM was called, because it would do the M&A normalization using SPM.
Okay.
So yeah, just use import anatomy folder. Then you select the anatomy folder that is in the workshop LA folder. Open. After we select 15,000 vertices, and we click OK. It will load the MRI and it will open the MRI viewer. So it's everyone in this point. Right, so what we're going to do is now to set the fiducials. The fiducials that were used for the recording. So what we can do in this viewer is we click here in this crosshair that is in the bottom. And with that, we can go direct to a specific point in our MRI. Those are printed in the guide. So for the nascent, where we want to do the MRI coordinates 125, 230, and 64. We click OK. And this point was the one that was used for digitizing the sensors. Once we are there, we just click in set in the bottom set in front of the nascent. So we just set. And we do the same for the other two fiducials. We go over here, we set the coordinates, the MRI coordinates, the ones that are printed.
But how do you know if it's Bayesian versus LPA or this is?
Oh, here, what you will see NAS, LPA, RPA.
But in actual practice, you wouldn't know those coordinates in advance.
Yes.
So in ideal case, when you collect the EEG data, you need to know where you digitize the nation and the two fiducials.
So you would just navigate to like I am now, and like I would just say Bayesian, even though I didn't.
So yeah, we're common to use this value in order to get the same value
Same.
later on.
I see.
But in your case, later on, if you have your
As well, you can see the coordinates for all the fiducials at the same time. Like you right click on the MRI, edit fiducials, MRI coordinates, and it will open a window with all the
Okay.
So it's possible to edit it from here as well. The advantage of going to a specific coordinate like we did is that you see where you are going to place the fiducial.
Do that again, or let's hear from
Once you have set the fiducials, you can click on view just to go and see that the nation should be over nation, left ear, and the last
right.
and click on save. What it's going to do is gonna save all those points in the anatomy, and as well, it's going to import the surfaces that were completed by CAT 12 that were registered to this anatomy. So
Did I just?
I'm showing things.
And you.
So now is it normalizing it to the M&I or what? It's more of a M&I.
it was already what I got was like, yeah, it's important with the information to do that normalization.
Yes,
Mhm.
but that was already done
That
prior.
was done.
Okay.
For now, what it's doing now is importing all the surfaces using the fiducial.
Got you.
Yeah.
Okay.
Sure. Yeah, because from David or import only the MRI.
All good here?
Yeah. Right.
Question. What was the processing done previously?
We'll put it there. But it's something that I want to go to label where
You can actually run any of that.
Yeah, MRI.
Yeah.
No, this is just like all of them.
No, it's only the.
I was just kind of curious what you said.
The same.
Prior to looking this up.
Yeah, because.
No, I don't know.
They are not for small. Yeah, I think that, yeah, I'm familiar.
That makes sense.
Okay.
Alternatively.
Oh.
I don't like it, but they.
Oh, yeah. Good to know. It's,
Like I have a mean that I want to be.
Yeah.
Those would be additional. In the MRI.
Got it, and.
So, yeah.
So what we then will be. Start using the head and the tension. But that electrode will be co-registered directly in the image.
And.
Just double click on the So you see, I cannot get on the web.
You can see if you're using the same fiduciary.
It's not in the finals, so there is really problem.
Honestly.
Yes.
So when you have your surface, if you want to.
Display the coordinates. I know, I think that is
You can just.
image, right? But I don't know how I get it.
Close everything. And then there is this. Surface, just double click. If you hit last year.
Can I just?
Oh, change them now.
Oh, I see.
Because, yeah. Just right click.
Oh, I sorry.
And you have display, you have.
Okay, great, thank you.
This is the one I should close.
Yep.
Okay. Yes, this is not.
Okay.
Oh, these are.
Now that we have imported the MRI that appears here in the tree, and also we have other volume atlases. For example, we have the DAL screen window, we can double click it. Now we can show a volumetric atlas. So for each box or image, we have a result.
Oh, it's.
We can plot now the cortex that was imported. We can right click, display.
So I did.
We'll show here, we'll show the cortex that was imported.
Yeah.
Well, we can use the different, like, the mouse wheel to do so.
You can just
Rotate it. We can change to make them smoother. For example.
That's good? Yeah, super.
Can show the soul side. As well, we can see the cortical parcellations. If we go to the scout.
Yeah, you put
We select here the different atlases, for example, the basic Giuliani atlas. And it will show the parcellation, the cortical parcellations for that. Atlas. You know, into the scout and then you need to select here the atlas. Yeah, so, but this is only, this scout will only appear if you have a surface displayed. So,
Wait, what's green versus black color?
the green mostly indicates is the default surface that is going to be used for this object.
You already there.
As you can see, it has different surfaces.
Oh, so the green is the default one, and then you can switch to another one.
Yeah, you can
just show this.
Oh,
then you toggle down. Oh, gotcha, yeah, drop down. Let's choose one of them.
And at the end, we just click on these cross. So by clicking this cross, we close all the images, and as well we clean all the data that it's in memory.
Here. Here we are.
Unless there is question from the staff, you can move.
Okay, so the next part, what we're going to do is to import the functional data. So now we have imported our anatomy. We have all the files that we can explore. It's a file explorer, so we can do right click and explore them. That will be later. You have time, you can go through it. Now we're going to import the functional data. So the first thing is that we move to here, functional data. And of course, we don't have any data yet for our subject. So we do a right click. And we want to click here where it says review raw file. This is not going to, this is the thing I mentioned in the presentation. So it's going to be able to open the raw file, but it's not going to be copied in Brainstorm. So it's going to be able to read it from here. That's why it's called review and not import. And we select our data that should be in the same folder, the workshop underscore flags. And now in data. And we need to select here in the files of type. This is important. We select the MEG, the Yokogawa. And it will show only one file. We select it and we click on open. Going to read the channel data from the file. And it will ask you if we want to do some registration. We say no, because this is already, the data is already co-registered, so just click no. And now we'll have over, once it finishes. We'll have our helmet, where the helmet that is for the MEG's device, and all the dots that represent the EEG sensors.
So the Yokogawa file, was it inside the subfolder in data, or was it?
It's inside of
Subject 01?
It's just data, and it's the only file inside of data. But it needs to be the workshop underscore.
Right.
Yeah, because there are sometimes people is using the folder inside Brainstorm DB. Brainstorm DB, you should not touch it because that's the database for Brainstorm. Any change that you do there, you may corrupt the database. Yeah, I think it's the wrong folder. You are.
Inside data. At the another.
This is the update. Thank you. Off now.
So when
What we're gonna do is change some of the channels.
So this might
So
be
we have a channel file that is like electro. So we will right click on it. I did channel file because we want to change some of the, most of the electrodes were properly read from the data in the type. There are channels like the 208. 208, that is detected as EEG, but it's not, so we can just change the type to EEG. Say for 209. And also for 231, that is E, not even sure what it's here, but we just change to miscellaneous. And once we have done the changes in the channel, we just close the window and it will ask us if we want to save the changes. Yes. I know what we want to check is the co-registration, so it's the same file in the channel file. Right click. Then we go to MRI registration. For example, we start with the EEG. Just go check. And those are, we want to see the labels. We do right click. And it should be channels and display label. Also, it works with control lead. We see that some channels, like for example, this one, F4 is not really in the scale. Yeah, and there are other channels that are a bit even under the scale. So yes. So in the channel file,
Where do you see underneath it?
there's the ones that are above C4. Okay. And then we start to move. At some point, there are like this F3. No. It's nice.
Sorry, what from the key channels? You right click on key channels and then what?
MRI registration. And the one we want to check either EEG or MEG. In this case, we're seeing EEG.
Opening this.
Like this one, it's like D09 that you asked me. Oh. So what we want to do in order to improve the location of the EEG sensors, we want to project them to the skull. So we do again in the channel file, right click, MRI registration, EEG edit. And we want to make click in this button, that's this button over here that says project electrodes on surface. What it's going to do is just to move the position of the electrode to the closest point to the skull. We click on it, and then it fixes the location of the electrodes. And we click OK. We want to save, yes.
Once
Okay, we have now checked what we have done is import the raw data. We have checked the location of the sensors with respect to the anatomy, and things are good. So what we want to see now is the signal. So now we see this. We have this icon that is also and it says raw. So we right click on it, and we can display different things. Like for example, we can display the MEGs and display time series. And this is almost all the time series from MEG. about this overlap. So here we have the record tab. So we can click here to show it in the separate. All the MEG. Here we're selecting all the channels. So we click all. We can select a subset of channels. In the case of MEG, for example, only the frontal left sensors. Again, like we can move in by page. So maybe slow down. We can see, for example, how much data we're displaying. This case is 10 seconds, but we can change it to any other. Again, in the we can change here the gain with the I'm here in the bottom right part. We should change the gain. Reduce it, of course. Once we can close the figure and replace the memory with this X button. We can plot as well for EEG, display time series. Same idea, we can change different channels. We can change the time. As well, we can display several signals at the same time. If we go to EEG, display time series. And one of the things that you will see in Brainstorm is that all the plots that have a common features is shared. What I mean is that, for example, here the time is always shared because the time series have a common thing is the time. If we do some in one figure, it changes the time definition in the other figures, which is very efficient and useful, for example, to
That's the
let the
plan. I wanna do that.
and they happen at the same time. We can we don't have oh yeah, we're supposed to have in the channel. So in this channel, we have the channel 214 ECG. Oh, yeah, we can double click the type and change it to ECG. Close here. Yes. All of this to be able to, now we have the ECG here, display time series. And for now, what we can do is to plot the EEG and plot EEG at the same time.
You're getting there. Yeah, here's another option for. So. You just need to check. And then you'll be done.
So basically different types of channels have to be displayed in different windows.
They will be displayed. It's possible to combine them in the same one. For example, people doing a sleep study, sometimes they want to plot different modalities in one. It's possible. We don't have the time to show that today, but yes, it's possible.
But if you name all of those the same, let's say you name your EEG and your ECG type as, and then you can.
Yes, but that's a very bad idea.
Oh yeah.
Yes. What you want to do is to create a new montage that you say, bring this EEG channel, bring this EOG, bring the ECG. Yes. May have seen as well that, for example, for EEG, we can do EEG, and there's not only time series, but we can display, for example, the 2D disk. There will be the topo plot. And again, because the topo plot will share as well the time dimension with all the other data we have. If we select different time, we can see different. When selecting different time, you can have a different topo plot.
So what topo plot is this? Topo plot of what?
EEG.
Like the amplitude?
Yeah, it's only the EEG presented
Right.
in the scout. Yeah, there isn't anything for some construction now. It's just the data in the scout.
It's just based on the post.
Yes, yeah, it's voltage. I'm not sure if it's visible, probably because of Zoom, but let's say it's microvolts.
That's what I'm saying, it's not, it's.
Yeah, no, no. No.
But it was the 16 side.
Instead of for the file types.
You can have it out.
Sign up file that have, like, for example, here we have our, and it's totally different. Like, ours, I that it's, that's, you know.
Think about, I think I do one.
Files.
We will need their. Just one thing at a time. So this one.
Money for ATL changes. We don't want to.
Process two.
Process, one thing. Instead of like.
Ah,
okay.
We have a trigger for.
Okay, all right.
We have the type. We just close the figure.
I'm just checking.
A very important step to do it for. To put the figures, otherwise we won't be able to find.
Yeah.
Well, the higher the.
I wanted to show how you can see.
Yeah, we have done the.
No, good.
The report. So let's start with computing the PSD. So we drop this file. To down here to the area where is the process. We need to be process one. Because we're processing only a file independently. We don't want to process two files at the same time. And we click run. We select the process. And the process we want to do is PSD, therefore it's inside the frequency menu. And PSD using the Roach method. We want to analyze all the file in windows of 5 seconds with 50% overlap. As units, we need units square over hertz. And we want to process MEG and EEG. We don't need to pre-process EOG or other signals. And we just click okay, we get up to here and we click on run. And yes, we need to check that the PSD options. The PSD options, we're using the MATLAB FFT defaults, okay? And then we can click run. It will create a PSD file that you can see that is nested in our profile. To display, it's the same idea with all of the other files. We can double click or we can do right click and then show, for example, the power spectrum. Power spectrum, we have two big groups. One is the EEG and one is the MEG. And mostly the difference is because of the MEG is going to be measured in femtoteslas. So that's why it appears that way down in the dBA scale. Well, EEG is going to be microvolts squared. And from this, there are a few stuff that we can start to appreciate. Like we do zoom in, we can notice a peak around the 10 hertz, that is the alpha waves in both modalities, in EEG in the top and MEG in the bottom. As well, we can see like on power line noise, we have a 60 hertz, we have a 120. ish, 180. And there is one sensor in the case of MEG that we can notice that it's a noisy sensor just because of the power spectrum. Otherwise, we can plot the time series and see that it's going to be very noisy. Yes.
If I go back and change the montage, so then I have, let's say, differential channels. Then this PSD, how does it affect it? Is it going to be replaced with the PSD or?
No, the montage is only for visualization.
Well, but what if I want to get the PSD of FP?
What you will need to do is to compute a new file that is actually the difference and compute PSD on that file.
So these PSDs are just the raw data that's inside the file.
Yes. Something what we can do is to select the channel so it looks in red and then we press enter. But it will open a new figure that it says with that data on it. So that is the LC11 channel.
Yeah.
And also put the topography.
Yeah, we can do as well. If we do right click, we can see it
From here. You don't get those markers.
Oh, yes, because it's not available. Yeah, we can select the topography like in this PSD with Control T. And because it has two sensors, it will ask us which one we want to plot. Let's go with MEG. And we can check the topography for different frequencies. And we see that this MEG channel is always bad, regardless of the frequency. So just one thing so we can select it and then right click and mask selected as bad. So we just make this channel to be labeled as bad.
That should be.
We're going to apply filters now. So in order to apply the filters, we are going to do it in pipeline. So we bring down the process. We run. We go to the pre-process. Notch filter. We want to notch filter for MEG and EEG for 60, for 120, and 180. Yeah. We don't click run yet, so we can start to do piggyback. So we go another process, and the process it will do first the notch, and after we want to do the bandpass filter. We can do the bandpass filter again for MEG and EEG.
Now I should see notch.
From 0.5 to 200 hertz. Yeah. And now, if you see now, we have two steps in the pipeline, the notch and the bandpass, and we can add more. So now we're like, do the notch, do the bandpass, and after go to frequency, and after all of that, compute the PSD. Once we click run, what is going to happen is going to run these three processes together. Well, not together, but sequentially. And of course, we need to check the advanced options for
Why do you need to do that? Because we're basically not doing anything. This is
yet.
okay.
Yeah, we need it because they're mostly in the connectivity functions. When we are using short time Fourier transform, we need to define as well the window length, the overlap. Yeah, that's feature was introduced probably like a month or 2 months ago.
Wow.
We still need to see it as with feedback from the users. We really need it. So when we click run, and it will do the notch, it will do the bandpass. Okay, it has to do the PS.
I made some mistake. Instead of running the right link removal on.
Yeah.
I was running on this and here it was going.
I think yeah.
Yeah. No.
So yeah, we have done it really continue processing to do.
No, but when you're trying to
EEG. So we close
On the filter.
all the figures. And then we plot the EEG time series,
Because if you click on
EEG display time series. And then we go to the artifact menu that is here.
it's better to do it
It's in the record tab. Then artifacts. Re-reference EEG, and we want to re-reference to the average.
I usually do that.
And of course, we want to work it only for EEG channels.
Because that's how
So we
we give
just
an idea.
write average
Okay,
and run.
irrespective, I think
Run and we just say because what is going to happen, it doesn't use the average referencing of the data, but it just saves the transformation, the matrix transformation to do the average. The average to be referenced.
And this.
Yeah.
Yeah.
You have to.
It's on the right. What did he say about the average? It doesn't average the data, but it.
Yeah, no, we have the ECG.
So when you visualize your
I know.
Try it. To me, it's like.
All right, so now we're going to do artifact removal. So we close all over,
like SVM.
we close all over figures and we open the EEG, but we open the one that is already not a bandpass.
Like you actually.
EEG display time series. And we displayed as well the time series for USD.
Yeah, of course.
What we're going to do now is to find all the blinks. So we go to artifacts. And we want to detect eye blinks. In the channel, it's in the EO2. Yes. In the EO2 channel, we want to analyze all the file. And we just hold, we want to hold this.
Actually, that's very easy. I didn't realize that was sorry. I forgot that we're already like over.
It will detect now the link, so we can, for example, check different events. And we can see the links in the EEG channel here and their influence in the EEG. Here you see the different instances of events. Again, we have this other kind of blinks. Well, we're going to merge all the blinks together. So we go to events, merge groups, and we call it blinks with S. So now we have 45 events of blinks. We want to do something similar with ECG. So we go to artifacts, detect heartbeats. Those are in the channel ECG.
They're gonna hurt.
We call it cardiac of the branch. In order to corroborate that those are ECG, we can plot the ECG signal. Right click, ECG display time series.
Oh, because the first time we did it here.
Right click.
Yes.
Sometimes we don't have, channel, and we don't have, and we clearly have an EEG artifact, and we just remove it without having the right EEG channel.
Yes, because what you can do when the heartbeats.
Oh, I
Where
see.
is the channel? You can use the channel that is the noisiest.
Oh,
And the blank.
So that would be ECG channel.
What it does.
The same thing with EEG, sometimes you have FP1, it's just blinks, like, oh, that's my, oh, now you're EEG channel.
EOG channel.
Yeah.
I needed to take.
Yeah.
Uh,
All right, so.
That is something.
Events that are.
And like I see it from here, but still like I agree.
Cardiacs with links. We can,
We are not there.
Yeah. Sometimes.
So yeah, at this point, we should have. To perform the ICA. Yes. So now we can make this. Just see, you go to artifacts. I'm going to compute a component. We want to use it for the entire file. Only for EEG. That's important. We're going to use the picker method. And we want to sort the components or the ones that are related to artifacts. That will be EEG and ECG channels. And just
It was.
that's it. Just click on run.
See.
And it will run ICA using Picard, which is quite fast, probably 1 minute. Yeah.
I think it was. We'll have to. Yeah.
And then this will open the active projection that we have for our data. So let's just plot some of the components to see what they look like. That's why I like.
Well, increase it again.
And we select the display component type series only for selected components. This plot, the first thing we need to do is to disable the auto scale. Once we are doing the components, they don't change of size. Yeah. And in this other window, we can start to I would clearly see that the first component is related to ECG. So we can just remove it. And we can see as well that for the same for channel, we can see the properties.
This one is.
I think that was really good.
I think it's finding the.
Why don't you pull that one? Yeah, selecting the components and after in this. We can see as well that the components more in this case more likely green that is related to your genes selected.
Yeah.
And we just click save. Now we're going to do the same thing for SSP and MEG. We close it. The channel, we plot the MEG time series. We go to artifacts, SSP for I blinks.
Yeah.
And now over events are called blinks. So we need to write blinks for MEG.
Okay.
Click on run.
The channel.
Yeah, because, yes, that I already see it, but I-
And now we select the blinks SSP. So you're now it's just simple plot the first.
but I don't know, I.
Yeah.
Time series.
Okay.
Yeah, sorry. The other one.
Topology and then just
Yeah.
select the first component that's
Yeah,
related.
Yeah.
So the first one you should keep on. The first one is the. Covered.
Okay.
Check.
Oh, okay.
Yeah.
Okay.
And the second one is the. And the second one is the SE you just did.
Mhm.
So you can click on the like topography to see the topography.
What is
The
the?
circle.
Oh, this one.
Yeah. And then you can. Squiggly line to see the
Oh.
time series. I suggest one.
Yeah.
So the first one. this is-
I want to get yours.
I'm asking, is this for blinks or for card?
Blinks, right? Yeah.
So the first one are very good markers, right? That's
Mhm.
not really.
Yeah, no, I think
The second one is
I don't.
what I would consider,
Mhm.
I know
Mhm.
and you can actually verify that. I think.
That
The
it's like,
thing-
someone has a
the
sort
one
of can handle.
After the circle,
But I don't know
and
anything about
after that, it's kind of like a squiggly line.
But,
That could be
uh,
the time
Like I
series
still think it's cool.
of
I think like the
projector.
only thing that I'm worried about is more just like.
See it like
So it's not.
Roll
like,
to a
probably
plane,
not
it should
sure, like, I'm
look
sure
like
the data is like super cool. It's like kind of small though, like. But I guess.
Oh.
Exactly.
Okay, got it. I don't
If
know
you
what
wanna
is-
simplify it, you can
Mhm.
close this. The one
This
that you
one?
just- Yeah, the one that you just
Mhm.
opened. And on the sides, it's all like the first like 10,
This one,
Yeah.
alright?
but the first 10 like. those,
Oh, sorry, you can.
Yes.
Oh, I see, okay.
And it
Oh.
should only be about like 10 seconds.
Oh,
Yeah,
okay.
And then if you scroll through the time series, eventually one of them will be like, so you can actually.
And then like.
Oh, sorry.
And then, blue.
Uh,
The one that just.
this one, okay.
So I think like.
So with the ICA and the SSD videos we need for pre-processing. And now we're going to import our data. We completely the wrong one. Up to now, we can see the data. Because it's a continuous file. What we want to do now is to work off our data based on the events that we got.
Oh, I have a.
We go to import.
Yeah, I reached out to my.
We want to select all the data. We want to use events for to do it.
Now.
And the time we want to select from minus to. What is it? Listed from-
Like, whatever, I don't know.
And select from minus 15 to 50 milliseconds.
Really, I heard
We want to
like
apply
rotations
the
are like.
projectors that we have done.
Because
That it appears there is more
I'll talk to
in the
them.
average EEG. We want to apply.
And
We
then
want to
one
for ICA, we want to go for SSP.
I wonder how to figure out.
We want to apply all of them. We want to remove DC offset, which should not be necessary because we already did the band pass. And that's it.
Okay. Yeah, I was kind
With this options. Fork.
so now.
And so after that one ebook, I'm sure if we want to ignore it, yes, ignore it. You will see.
I think that was.
And the file that it was.
To-do list. So, yeah, she's gonna double check right now, actually, I heard that.
Okay. And then.
I had talked to them.
Yes, and then.
I think it is. And you have to right click.
Oh, of course.
That's it.
We're expecting some
Not have
some evoke responses on the left motor cortex, so we can go to an electrode over there like C3.
I went to his lab meeting a minute ago. It was really nice, but it was like
Now it's here. Now. And you have your
Like
IC.
somewhere.
I removed them.
It's like I actually went and asked, he's like, oh, yeah, like I really want to see you 2 semesters and like scan picture in the lab. But so can you rotate
It's
it?
basically it's grayed out because this ICA.
It's wrong here because I imported from epoch. I imported only from. Yeah, I didn't import the entire.
Okay.
I imported from 0.5
And then I reached out to.
from 50 milliseconds, not minus 50 milliseconds.
Delete everything, right?
So yeah, we can just remove it with the link here. That's it.
Yeah. But I
Nice.
know.
I think doing some mistakes, we learn. We learn.
And there is a minus that was missing.
When I figured it out.
There's
But-
They have
There
this.
is, because I think that there is. You have the dial. That's the time.
I don't think it's gonna make a difference. Like it or not, so.
Yeah, actually send it to me.
I think that's more useful when you have like. From all.
Like how I think.
With all this data now, we can start to compute ERPs.
But.
And then
have to make my full.
potential for EEG and event related field for MEG. So we drag and drop all the files. So we can do it like this. Okay, we want to run the process. That we want to do the average. So we want to average all the
Emailing Paul Thompson.
we want to average everything with only arithmetic mean.
It was just one, I sent him like,
We run it.
like before school started, and then when school started.
And now we
Like,
start.
I wasn't like.
ERF. So we do right click, EEG, display time series.
That's really close.
We can choose, probably now we're more interested in.
I wanna figure out how we're stage. It's so sad.
We do the same for energy. Playtime series. We can display the topography for both, like right click, view topography. Right click, view topography. And we're expecting around 20 milliseconds, the first the component, the N200. In case of people working with EEG, it's quite common to see the negative in the upper side, so there are these options to change the polarity. Yeah, that's so now it looks more closer to what I expect from the literature.
He said, can you come on
We can
Tuesday?
zoom in. And if we show the networks, for example, the network is right here. Channel display, channel labels.
It's here.
Once you segment the data, you can.
So yeah, we can have around.
Like, that's why I talked to her about it. That's how my concept would be like.
Here it doesn't matter.
Yeah, because we only have one.
Exactly.
Right? Okay.
So not touching that. So. Really? It's accurate. No, I mean, I actually prefer.
All right, so once we do the averaging with the files, we can display it. So with right click and display the time series, and right click and display for EEG, and right click to display the to the disk, or from the time series right click and view topography. So we should be able to see these plots for around the N200 components that in the ERPs. Right, so now this will be part of the analysis that we did at the sensor level. Next step is to do the source estimation. So we pull all our plots. And now the first thing that we need to have in order to compute that is the forward model. So we can go to the anatomy view, because we're going to create the boundary elements method surfaces. So we do right click in the subject one, and we want to do MRI segmentation to obtain the BAM surfaces. We're going to use the Brainstorm method. Here, usually we want to have 1922 vertices. However, for sake of time, we're going to reduce them for the number. The numbers that are indicated in the sheet. There is 1082, 642, and 642. We give still 4 millimeters for skull thickness.
Sorry, where did you get these numbers again? It's like the 1092 or the.
On the sheet?
Right, no, I mean,
Why these numbers?
yeah,
Oh,
exactly.
yeah, well, we were doing the smaller numbers because of the computation time.
Right.
Otherwise, it will take probably 10 or 15 minutes to compute the forward model with a high degree of detail. Uh-huh.
Okay.
So that's, but normally I would.
Now we will over,
Like when you.
we want to fix over the surface because beforehand I know it's gonna have some
Yeah,
troubles with
yeah.
some vertices.
Right.
So we do right click.
Very specific.
We force to be inside the scope, inside the cortex.
Oh.
This will not happen if we use the full resolution for high resolution for the bent surfaces. So it will just create a new surface that is called underscore fix, that it has all the vertices inside.
So this step might take some time, so we are going to use lower resolution.
Yes, we will.
Yes. It may take 5 to 10 minutes, but it's too long. But in real time, once what high resolution, we're going to use very high resolution context. It may take maybe 1 hour. It's fine. Just for the photo. Pretty quick. That's not like that. Lower. Yeah, it's very fast. But we can use the spherical head model and it will take few seconds.
Yeah, well, return show that.
Yeah.
Fears are a very.
And you can show also the FEM if we have.
Yeah, well, the FEM and the same thing, we can, with the, with the BEM surfaces that we select the BEM surfaces and right click. And we can generate now the FEM meshes. I think it,
They don't have the information.
yeah, in this case, I'm just showing it, you don't need to do it to generate the FEM mesh.
You have to do the BEM mesh first before the FEM? No, it's independent. Either you can do or solution or FEM solution, or both of them if you want. Just the mesh. Yeah, just the mesh. And now it's coming. It's just meshing the.
It was fast because the surfaces were already
Down.
sampled, yeah.
But Richard showed better image this morning for the head shape. You did it?
So now we should have our BEM surfaces, and we have also the cortex that has been fixed.
Yeah, this is the forward.
So we go back to, this is the anatomy part, so we go back to the functional. And we want to do is to create the forward model, so we do right click in the channel file. And compute head model. The head model is the forward.
I know from like.
We want to create it only for sources, we want to create it for sources. Models, one for MEG and one for MEG.
Yeah, so if you look here.
We just click OK.
Image is overlapping spheres.
Yes, because with images,
Oh, yeah.
You don't need regular B.
yeah, the advantage with the tissues are permeability, so the process is correct. Yes.
Yeah,
It's in the vacuum.
yeah, well, and earlier,
We just
MATLAB
computed.
crashed on me, so there's also the possibility that even though it looked like it.
Yeah, but in any case, the process is correct, so you still see localization of things. My guess is that.
I'm not.
We use for this cortex to be inside the BM. No, the default is the one that is in green.
I mean, right
Yeah.
now.
So, but you modified.
Process.
The process is correct.
not too worried. Because I think the main idea is just getting sense.
Yeah, I'm like.
This is the guy you want to talk about.
Differences in benefits and advantages of them.
Yeah, your own experience, but overall, the BM is very, it's a realistic head model.
Okay.
It's very smooth. If you go back to the sweepers, are very smooth as you saw there. And the version of the FEM that we are showing here is just a simple version of the FEM. But if you want to run very real FEM, you need to take the T1 and T2 MRI of the participants. You will do the segmentation, extract not 3 layers, which is just the inner outer scalp. You will extract the white matter, gray matter, CSF, scalp, and skin. You should have 5 layers model. And from there, you will assign the computer
covariance. So we select our recordings, right click, noise covariance, compute from recordings.
Yeah.
So you can take close to
We select them.
So the way
We're going to use all the data that is before the stimulus, so the noise covariance is not affected by
It's just a random idea.
So for minus
But you
50,
did
Pretty creative or done that.
right now,
we
it's
want to
going
do the noise
to
covariance for the capacity because we're going to compute sources for both modalities. We click OK.
There is some theoretical.
And now that we have the head model, that is the head model for the EEG, works in EEG, we can compute finally the sources. So we do right click, compute sources. We're going to use minimum norm, current density,
They just.
constraint,
There has been
and
some.
only MEG.
Years ago, someone did.
Can I just ask the difference between constrained and unconstrained and the other one is?
Yeah, in constraint, what it was all doing is all the sources were assuming an orientation, and the orientation is normal to the cortex.
Yeah.
Therefore, each of the dipoles that we're finding is only, we're computing only the magnitude. In unconstrained, we are assuming that in each cortex has actually 3 dipoles, and they are so for each vertex, we are finding 3 magnitudes for X, Y, and Z. So the
You're gonna get
dipoles,
2 different values?
yes, they will have different values, so that we can see that we're finding a 3-dimensional dipole. In this, some of the orientation will change. And in the constraints, I don't remember what, but I think it's like constraint, but you are allowed it to move a bit. It's not completely 3D. Also easier to interpret because on the other one it's harder to interpret when you have it.
No, I agree, yeah, I don't.
Yeah, sorry.
Alright, it's
We have about channel, that is the channel.
so I feel a little bit beat up for him. Yeah. Beat up.
And we do this again, the sources for.
So, but not interested.
But now this time we do it for EEG. If we try to select EEG at the same time,
Yeah.
it will tell us that Brainstorm cannot handle the 2 modalities at the same time for compute sources. We click OK.
We also tried.
Files, those are the kernels.
But if you.
Because what we is only a morning with Richard with the slides, what we're comparing is just the.
Oh, wow, it's almost like you're trying to sell this.
Sensors to sources.
It's almost like you're trying
So
to sell this.
Marketing.
Yeah.
So we only have here once. For example, here we have 300 and something files, so we don't need to compute. This will be a very heavy file, 300 times. That's why we only have something that is called a link. That's what it does.
I know what your friends were doing. Right. Because Kika's usually just right. So
The plan.
So,
but yeah, I'm sorry, I just like.
Just turn off the scan.
And we plot as well the time series.
This is only correlated to
Plot the time series.
Yeah.
20 milliseconds. We can change in the surface, the amplitude.
So I would have to be.
What we are seeing now is event related fields at the same time that we have the sources and the location of where is this activity coming from. That is in the place where it's pegging around the central focus in the left side.
Is the because it was
Hello. Just to finish the workshop, I want to show you that in the page with the data for the tutorial at the end, we have this link to one script, and this script will do this script will perform the different steps that we did today. You can. Mostly when you open it, you will see that it does the, well, you can probably not see that well from far, but it's imports the data, the anatomy, imports the recordings, do the steps for projecting the EEG electrodes on the skull, and all the pre-processing. And there was a question how we do this in Brainstorm, how do we bring it? For example, if we want to do the average, we just, like, we bring the files over here, do run, we do average files, and then we have this button over here that is for load and save pipeline. So we click on it, and we generate a MATLAB script. It will ask us where to save it. We save. I had a file before, and that's it. Well, it will save in this case, the name of the files that we want to do, and it will save the code with comments already, what is happening, all the variables that we need. And you can do that as well with all the, if you create a more complex pipeline. So if you start to do different processes, like pass filter, and after you have, probably can do it with one file, so it's shorter. We want this file. I want to add a notch filter and pass filter. After we want to create the PS, we're going to compute the PSD, same thing. Save as, sorry, generate a MATLAB script. Yes, we need to check the options. And that's the code. So you will see have the block to compute the notch filter, the block for the bandpass filter, and the block to compute the PSD. Yeah, we just need to click run. Well, we can do it.
That's very cool, isn't it?
And I think with this, yeah, we conclude the workshop today.
So before we wrap up, I want to thank Raimundo, Katrinas, Chinmay, and
Jason,
you,
Wujay,
and Wujay.
Oh, it's here. You just changed the sweater.
So thank you all for running this.
Yeah. Thanks, everyone.
And I hope you'll all be here tomorrow. Change of pace. The EEG machine learning symposium. We have some really interesting speakers, and we start I think at 9 tomorrow. And I believe we're providing breakfast again and lunch. Yes. Oh, and of course, thank you to Gloria who arranged all the stuff, and Mickey and Ariana, Seth, Mayumi, everybody. So have a good evening. See you tomorrow.